So it's the top of the hour, and I'm going to start. Welcome to the 2021 Charles H. Tom Sawyer Memorial Lecture. Tom was a friend and a mentor and a colleague for some of us, and I realized that with the passing of time, the number of people who actually knew Tom directly is shrinking, and so it's important to, I think, give a little bit of an introduction and some context of why we honor Tom with a memorial lecture. Tom was a towering figure in the field of neuroendocrinology. He began to study neuroendocrinology before there was a neuro in front of endocrinology. His work established the importance of the brain in the control of reproduction, both the behavioral aspects and in females, the control of ovulation. And here are three papers that very early papers that kind of define Tom's career. And you can see from these papers that these are the fundamental questions of neuroendocrinology. In the first classic paper with Everett in 1950, they first established that the luteinizing hormone release apparatus was really controlled by a part of the hypothalamus, which at that time was not really clear. And we are still working on understanding both the connectivity and the actual neurotransmitters that are involved in this luteinizing surge that's important for ovulation. He also demonstrated early in 1959 that steroids, in addition to acting like hormones, had electrophysiological properties in the brain that were reminiscent of neurotransmitters. And this work was revived around the turn of the century when we discovered that there were a series of receptors, steroid receptors, that are found on the membrane of a number of cells, in particular throughout the nervous system. And finally, Tom, never shying away from the big questions, was interested in this mysterious aspect of development, which we call puberty, and which we still don't really understand how it's triggered, but we understand its crucial importance in the development of the individual. And Tom trained a whole generation of neuroendocrinologists. They were postdocs and graduate students in his lab, and a number of his colleagues, his contemporaries, were big fans of Tom's. And I want to read you two quotes, one by Roger Guillemin and the other one by Andrew Shalley, who shared the Nobel Prize in 1977 with Rosalind Yalow. Roger Guillemin and Andrew Shalley agreed on almost nothing, except that their praise of Tom Sawyer. And Roger said that Sawyer was one of the very early contributors to the field of endocrinology through his fundamental studies of the function of the ovary and the brain mechanisms now known to be their ultimate regulators. He always asked the right question or provided a clue for the right answer. And Andrew Shalley, who agreed with Guillemin about almost nothing, said with deep admiration and friendship to Tom Sawyer, the #1 pioneer in neuroendocrinology and the man who started the avalanche of progress on the hypothalamus. Tom, in addition to loving research, which he did for decades, was just an absolute fan of teaching. And he gave actually the first lecture in the newly formed medical school at UCLA in 1951. So Tom taught gross anatomy, which he picked up on at Duke. And here he is dissecting a cadaver with some students. And he always used the latest pedagogical tool. And I love this slide. And this is Tom using 3D glasses in the 1970s among a group of medical students. And as all good academicians, Tom did some administrative work. He was the chair of the anatomy department way back when, which now is the neurobiology department at UCLA. In addition to his dual loves of teaching and research, he loved to go to international meetings. And I think part of this was because he enjoyed meeting with people throughout the world, some of which were his trainees and others whom he had met because of their interest in the same things that he was interested in. And to honor that, the Department of Neurobiology annually gives a postdoctoral travel award in the name of Charles Sawyer to enable someone to attend an international meeting. Tom gathered around him a group of faculty, and this neuroendocrine group became the Laboratory of Neuroendocrinology. And the Laboratory of Neuroendocrinology was initially headed by Roger Gorski, and then Art Arnold, and then I am now honored to preside over this group. And here are the current members of the Laboratory of Neuroendocrinology. And it is the trainees of these laboratories who each year pick the Sawyer lecturer. So it's a great honor, I think, to be picked by the trainees. And this year we are welcoming back to UCLA our friend and colleague, Larry Swanson. And it's a pleasure to welcome you back to UCLA, Larry, even though it's virtually. Many of us, have interacted with you, are your friends and your colleagues, and some of your mentees are here, including in faculty positions at this point. And so it's a great pleasure to have you here. Larry began his academic career at Pomona College and then moved to the Washington University in St. Louis for his Ph.D. and did postdocs with Max Cowan and then the laboratory of Rita Levi-Montalcini. He returned to California, to Southern California in 1980 to the Salk Institute, where in 1985 to 1990, he was a Hughes investigator and then joined the faculty at USC. Larry, I've never understood why he didn't come to UCLA, but that's a different issue. I met Larry in the 1980s, and he was busy dissecting the paraventricular nucleus of the hypothalamus, which kind of I've always viewed that as Larry's test case for his later work, that the most important thing that we needed to understand was the architecture, the organization of the architecture of the brain, if we were ever going to get a mechanistic explanation of various brain functions. From a personal note, those of you who've never read a Larry Swanson paper, I really recommend the experience, because in addition to exquisite experimental work, the scholarly nature of the discussion is very, very enriching and very, very educational. You'll learn not just about the area Larry examined under his microscope, but the history and the current state of the literature for that area. Larry has been extremely prolific over his career, publishing over 200 papers, some 70 chapters and reviews, 5 books, as well as the translation with his wife of 3 books of Ramoni Cajal, and co-edited an additional 7 books. And the list of honors and awards that Larry has is truly exceptional. And in this case, we just can't enumerate all of them, otherwise Larry won't have time for his lecture. But I would like to hit some high points. Larry was the president of the Society for Neuroscience. He was the president of the Cajal Club, which only means anything to neuroanatomists, but it's an important honor. He was Secretary General of EBRO. He is a fellow of the American Academy of Arts and Sciences, and he is a member of the National Academy of Sciences. And so without further ado, I'm going to stop sharing my screen and allow Larry to introduce his lecture today on understanding the basic plan of the nervous system.
Paul, I can't thank you enough for that overwhelming introduction, really. I sort of wish my mom could hear that one. That was great. I was really surprised and honored when you asked me to give this lecture, and I can't thank you and the Laboratory of Neuroendocrinology enough for the chance to talk to you today. It's really, really a deep honor, and I really appreciate it. So what I want to do, let me start off actually with a couple of anecdotes about my interactions with Tom Sawyer. So let me just share the screen here. Hang on a second. Okay. You know, I think I met Tom first in the mid to late 70s. I'm not exactly sure when, but I met him and talked to him. And Paul, one of the things I asked him, I said, you know, I would love to get a job at the Brain Research Institute. And are there any openings? And he said, well, we have an opening, but we're taking somebody from the inside, but I'll keep you in mind. And actually, that's the last I ever heard of that. But I came up from La Jolla in 1984 for this absolutely wonderful symposium celebrating the 25th anniversary of the Brain Research Institute. You know, as you mentioned, Tom was one of the big five that started that, along with French, Lindsley, Magoon, and Ted Bullock. And this is actually a great book outlining the first 25 years of this wonderful institute. And Sawyer was nice enough to sign his photograph in the book there for me. As you mentioned, I mean, there are a number of things that people remember Tom for, and I think primarily his science. And when I started grad school in the late 60s, my Bible really was this book by Haymaker, Anderson, and Nauta, The Hypothalamus, which was published in 1969. And Sawyer had this classic chapter 11 in this book, Regulatory Mechanisms of Secretion of Gonadotropic Hormones. And I was actually lucky enough, this is his copy of the book. Russell Johnson had a silent auction down there in the reading room of the rare book room of the Darling Library, and I got his copy of this wonderful chapter, which, if you're at all interested in the history, you should reread this. Another thing about Tom, you know, was this incredible group of students and fellows and visitors that he entertained. And this is an absolutely fabulous picture. I think it's probably an international meeting in Mexico, but you know, Sawyer on the left there with actually his first postdoc, Charles Barraclough, who joined him in 1953 and went on to great things. And one of Barraclough's students was Roger Gorski over there on the left. And I got this picture from Rich Simmerly, who was a student of Roger's. But it just shows the collegiality and wonderful times they had at the Brain Research Institute in the early days. And then finally, of course, I came up to this memorial celebration, really, at the Faculty Center in 2006 for Tom. So he's one of my heroes in science. I have always admired him for a lot of reasons. So let me now get into what I wanted to talk about today. And, you know, back in grad school in St. Louis, I was studying the control of ingested behaviors, eating and drinking behaviors, by injecting neurotransmitters into freely moving rats, injecting around the limbic system. You know, for the students in the audience, this is not optogenetics or anything fancy. This is the old school of polyethylene tubing with a cannula implanted in the brain. But the really interesting outcome of all of this that was really articulated by Pete Grossman was that there are hypothesized that there were neurotransmitter coded circuits for different classes of motivated behavior in the brain: eating, drinking, reproductive behaviors, defensive behaviors, and so on. And so I really became interested in what are those neural mechanisms, what are those neural circuits. And that's really what I've been working on ever since, and really is the background for what I want to talk about today, because after all of this research I've done over the years, I mean, it's become more and more obvious that to explain any behavior, any naturalistic behavior, the biological mechanisms underlying those behaviors really involves understanding the entire central nervous system, the organization of the nervous system. Because after all, any of the sensory modalities are involved: foraging behavior, memory, satiety, motor activity, on and on and on, which in essence is the nervous system. So just by the research that I've been doing, I've been sort of forced into this conclusion in this line of research that I've sort of taken up in the last 10 years or so. And so for perspective, for the first part of the talk, I mean, what better perspective could you get than somebody like Einstein, who in writing about the evolution of physics wrote that, "Let no one suppose that the mighty work of Newton can be really superseded by this or by relativity or any other theory. His great and lucid ideas will retain their unique significance for all time as the foundation for our whole modern conceptual structure in the sphere of natural philosophy." You know, I think there's a tendency these days to say if something wasn't published in the last year in Cell or Nature Neuroscience, it's probably not worth looking at. But as a matter of fact, there's a very, very rich, substantial history in neuroscience that really shouldn't be, the foundations really shouldn't be disregarded. And so what I want to do today actually is almost go step back to some first principles about circuits and almost start at square one and try and build up an argument for a strategy on how we can actually get at the organization of the nervous system. And over here on the right, I mean, this is actually ultimately what we want to understand: the human nervous system, the central nervous system and the peripheral nervous system. What are the basic design features of this system? What is the wiring diagram? It's really the only body system where we don't have a clear, simple conceptual model. I mean, it's very easy to describe in general what the digestive system does. Harvey described what the circulatory system does in 1628. But when it comes to the nervous system, we know what neurons do. We have a great understanding of neurons, but we really, as a system, we really don't have the general principles down. We really don't understand it. This is probably still the best drawing of the human nervous system I've ever found, and this was from Vesalius back in 1543. So, I mean, there are some general principles for solving complex problems that are time-honored and tested that actually work for any complex system. And the way to attack solving a complex problem is that you need a parts list is absolutely fundamental. You need to know what are the elements, basically. You need work. You need to know how are the parts connected. And then you need to know how the parts are interconnected and work as a functioning system. And finally, there's a strategy for solving any complex problem, which is basically to proceed from a coarse bird's eye view explanation down to eventually fine grained solutions. In other words, start with the big picture and work your way down. So I think the Newton of neuroscience, at least from my way of thinking about it, was Cahal. And here was the basic model that he came up with for systems neuroscience, talking about parts and interconnections in a nested hierarchy. And he articulated this back 100, 125 years ago, but I think it's still the same basic way that we think about and teach neuroscience. And that is that the nervous system is parceled into different gray matter regions. For example, here he's showing the retina and the tectum, but there is the primary visual cortex, there's the suprachiasmatic nucleus, the lateral geniculate nucleus, the cerebellum, and so on. So at a sort of a macro level, the nervous system is parceled into gray matter regions. Each gray matter region has a unique set of neuron types, and that's obvious when you look at the retina, for example. There's a hierarchy of neuron types in the retina. The same applies to the tectum or any other gray matter region. And as a matter of fact, that's really what defines a gray matter region, is the unique set of neuron types that make that gray matter region up. And then each neuron type has a unique set of individual neurons. So neuron types over here could be rods and cones. They could be ganglion cells, bipolar cells, whatever. And then each gray matter region itself has a unique set of extrinsic and intrinsic connections. So if you think about a part like the retina, what you'd like to know is, what is the intrinsic circuitry? How does it work as a part? And what are the other parts that it's connected to? The inputs and outputs. So that's really a framework, the framework that Cahal gave us for thinking about how to attack the organization of circuits, actually in any animal. And it's the framework that I'm basically building on. So back a while ago, actually 2003, Hong Wei and Mihai Bota and I wrote a perspective paper that we called From Gene Networks to Brain Networks, where we basically laid out what are the fundamental problems that are blocking our attempts at getting at the basic wiring diagram of the nervous system, and making a case for having a very accurate, detailed structural model of the nervous system, like Vesalius provided for the human body back in the Renaissance, and Watson and Crick provided for molecular biology in the 1950s. We argued that we need an accurate physical model of the wiring diagram of the nervous system to start making progress. And the third thing we did in the paper was actually have a prototype knowledge management system for neural connectivity on the web. Two years later, Olaf Sporns and his colleagues published a paper that really shook people up, a lot of people up, where they actually introduced the term connectome and talked about the need for a human connectome. And they defined the connectome, a human connectome, as the connection matrix of the human brain, as representing an indispensable foundation for basic and applied neurobiological research. So a connectome is conceptually incredibly simple. It's a connection matrix. Here's a simple connection matrix with 10 nodes. They could be regions, gray matter regions. They could be neuron types, or they could be neurons. And the idea here is that you have, let's say in this connection matrix, 10 nodes, 10 gray matter regions, and it's a lookup table. You have this list of 10 parts by the same list of 10 parts, and you have a connection from a region of interest to a region of interest. And it really tells you what the connectivity of these 10 nodes are. For example, if your region of interest is number 7, if you look across this row, you can tell everything that it's connected to. If you look at this column, this region of interest, you can look at all the inputs to that region over here. So it's really just a lookup table, a way of organizing all the connectivity data about these 10 nodes. And of course, the power of this is that this is a matrix, that you can put it into MATLAB or any other analytical, mathematical analysis tool you'd like. It's a very, very powerful, seemingly simple approach to organizing all of the known data about a network. And you know, it seems very simple, straightforward, but look, here's a great table. You know, this is Mendeleev's original publication on the elements. And at the time, back in 1866, there were actually, you know, only 62 known elements. They arranged in these rows or columns. But look at the question marks in here, for example. And what he really created here was a predictive, testable hypothesis generator. In this case, with 6 columns and 19 rows. And look today what happened. I mean, every classroom in the world that teaches chemistry has got a periodic table of the elements. These are the elements, the nodes that make up matter. And when you arrange them in the correct order and so on and so forth, you start to get modules. You start to see relationships in there between these different elements, okay? And so it's a way of not only of organizing data, but seeing relationships between the different parts. And this is really the power of connectomics, which I'll talk about our preliminary results as we're coming along. And when you have a table, a connection table, the revelation to me, and you know, I'm new to this in the last 7 or 8 years, is partly, and I think this is a new way of thinking for a lot of neuroscientists. I mean, it's certainly not new to people that deal with networks and so on and so forth, but network analysis based on graph theory. I like this example based on airline routes throughout the world, projected onto a flat map of the Earth, analyzing the network of airline connections over a 24-hour period. And the whole idea here is that you have a very simple nomenclature for describing what's going on here. And in red, over here, these little dots are airports. So that's where flights arise and end. Those are nodes in the circuit. Connections are flights between nodes. So from to, directed flights, that's a connection. A hub is a node with a lot of connections. So down here in the Antarctic Peninsula, you've got some nodes with almost no flights. But over here, you've got LAX and SFO, which are hubs. They've got their nodes with a lot of flights. Over here is JFK, Heathrow, so on and so forth, hubs. And then there's a rich club in a network, and those are hubs that are highly interconnected with each other, much more so than would be expected by chance. The idea is people like Bill Gates and Sergey Brin are much more likely to know each other in a rich club than they're likely to know you and me. So something like LAX and JFK and Heathrow are part of a rich club in this network. And the final thing, of course, is subsystems. And you could think maybe there's a subsystem of airline routes within a continent, but there might be subsystems based on airlines like American Airlines or Air France or whatever. And so really, this is the type of network analysis that can be applied to any complex system, basically. And it's a nested analysis strategy, because you can take, for example, here an airport is just a node, it's a dot, but you could go in, you could zoom in on that airport and look at the terminals. You could look at finer and finer nested details in this whole system, but this is the macro level system here. So that's the basic terminology I'm going to use in describing how we are trying to get at the subsystem organization of the nervous system, the wiring diagram, the architecture. And so here's our overall strategy for getting at this, which we started 5 or 6 years ago. And here's the nervous system of the rat. Here's the central and peripheral nervous system, sorry. And here's a flat map of the central nervous system with the basic parts that are actually derived from embryology, from the basic divisions of the neural tube. And, you know, in the neural tube, you've got a forebrain, midbrain, and rhombic brain, which are broken down farther and farther. And actually, at the lowest level here, there are about 11 big subdivisions that are basically gross anatomical and embryological. And we decided to actually take a very systematic approach at getting at the connectivity of this incredibly complex nervous system by really just starting at the rostral end and moving our way down caudally. And so we started with the telencephalon, the end brain, the cerebral cortex and the cerebral nuclei. And then we did the hypothalamus and thalamus. And we actually did these together, and the thalamus and hypothalamus together, and then the forebrain. And so at this point, since 2015, we've published 9 articles on these major parts of the central nervous system, really getting at, to begin with, what's the intrinsic circuitry of the cerebral cortex? What's the intrinsic circuitry of the cerebral nuclei, the basal ganglia? What's the intrinsic circuitry of the cerebral hemispheres, and so on, forebrain? And here are those, whoops, sorry about that. Here are those parts on a flat map. And so I want to talk about now, we've gotten to the point where actually we've got the connectivity of the whole forebrain done. It was published in 2020. So you can read the details in there, but I want to go through the basic findings with you today, because it really, we're about halfway through this project, and there's a lot of very interesting things that are starting to come out of all of this. Although, of course, we're not going to have the final answer until we've got the whole thing done. But here is the connectome of the forebrain, the rat forebrain. There are 466 gray matter regions in the rat forebrain by 466 gray matter regions, okay? And as a matter of fact, this includes right and left. So there are 233 on the right, 233 on the left. And the raw data, we fundamentally went through qualitatively and provided ordinal values from very weak to very strong to connections that are present. This is all, well, I'll tell you how we did this in just a second, but this is what the raw data actually looks like. It's a 466 by 466 matrix. You can't really even read what the nodes are, the gray matter regions are here, but I'll show you that in a minute. So here are the basic features of this forebrain data set. It's a 466 by 466 gray matter region connection matrix, which means that there are 216,690 possible connections. That's really 466 squared minus 466. So even at the macro level of retina projects the lateral geniculate nucleus. Lateral geniculate nucleus projects to primary visual cortex. There are 216,000 possible connections in the forebrain. So reading through the literature, we found 375,000 connection reports for connections of region A going to region B. Now, what is a connection report? Well, Joel Hahn created a database, a conditionally formatted Excel spreadsheet each row in here is a connection report. A goes to B with metadata in the columns. And there are 19 columns of required metadata and 62 columns total of metadata. So this is a huge database of connections. A goes to B, et cetera, et cetera. And on average, we found about 2 connection reports in the literature for every connection. So here, this Excel spreadsheet for the forebrain has about 375,000 rows. So this data came from about 53 different journals since 1973, and about 276 labs were involved in generating this data, and about 26% of that data actually came from our lab. I mean, this is actually what our lab's been doing for almost 50 years. So in essence, we're almost taking what our lab and our collaborators have done over the last 50 years or so and subjecting it to network analysis. Now, out of these possible 216,000 connections, about 18,000 connections are present. There's data for about 18,000 connections out of 17,000. That means that this network is about 17.4% of all possible connections are formed. So in other words, about 23% of the, about 83% of the connections are not formed, are absent. So that's the connection density. About 17% of all possible connections seem to be formed. At least there's evidence for that. And on top of that, data was found for about 95% of all possible connections. This was absolutely shocking. I had no idea this was going to be true when we started out. And what it implies is that basically the rat connectome is already in the literature. Just about the entire thing is in the literature. And we've gone through and we've got a validity metric for the type of pathway tracing method that was used to get this data. It's a scale from 1, which is horrible, like HRP, to 7, which would be PHAL. And the average validity metric for the pathway tracing methods for these data on present connections is about 6 out of 7. So it's actually based on very good methodology. Now, something that's very important is that out of all of these connections, there are 4 very clearly highly sexually dimorphic connections in this data set. It's the right and left connections from the BST-PR to the AVPV and PMV in the hypothalamus. I'll get back to what the significance of this is a little bit later, but these connections were established by Rich Simmerly, actually, and means that we actually have to do separate connectomes for the male and female because the connectivity is different. And you'll see what that means a little bit later for the overall connectivity of the male and female forebrain. So the analysis of all of this was basically a method that Olaf Sporns and his colleagues came up with in 2018. called multi-resolution consensus cluster analysis. And I don't really have time right now to go into how this works. I'll be glad to talk about it in the discussion at the end. But it is essentially a very sophisticated cluster analysis of this connectivity. In essence, what is most highly connected with what? So this is what that connection matrix looks like when it is subjected to multi-resolution consensus cluster analysis. And what you can see, the beauty of this method is, first of all, it breaks all of that data up into clusters, which we're calling subsystems in terms of systems neuroscience, subsystems. And what you can see at a very top level, there are two subsystems, S1 and S2. But this multi-resolution consensus cluster analysis also yields a hierarchy of subsystems or clusters. And this is extremely important because it actually makes the data approachable, as I'll show you in the next few slides. So to start to think about this, here is subsystem 1. You can see subsystem 1 here from subsystem 1 to subsystem 1. These are the intrinsic connections of subsystem 1. Here's subsystem 1 projections to subsystem 2. Here are the connections within subsystem 2. And here are the connections from subsystem 2 to subsystem 1. So this is how you read this matrix. But then what you can see is that subsystem 1, for example, here, is broken up into two big subsystems, subsystem 1.1 and subsystem 1.2. These are the interconnections within subsystem 1.1, system 1.2. And this is bilaterally symmetrical, so you actually have the same thing going on the other side. And you have connections between the subsystems. Now, the other thing that we did for the forebrain was to start functionally annotating this hierarchy here. So this is really the structural hierarchy based on connectivity. What is connected to what most densely? But if we start to then annotate known functions on these clusters, we can begin to get a structure function subsystem model of the forebrain. And at the very top level, the forebrain turned out to be exceptionally and surprisingly simple. I was, again, absolutely astounded at this, but that huge number, that 17,000 connections in the forebrain, basically divide into two subsystems, two bilaterally symmetrical subsystems, subsystem 1 and subsystem 2. Subsystem 1 is the right forebrain plus the left retina, and subsystem 2 is the left forebrain and the right retina. And what you can see is that the intrinsic, when you look at just weighted connection densities, that's the sum of connection strengths divided by the number of nodes. The intrinsic connections are much stronger than the connections between the subsystems. So actually the right forebrain and left forebrain are actually very weakly interconnected compared to the connections within. In fact, that's about a 13-to-1 ratio. Now, it didn't have to turn out like this at all. For example, we just finished the midbrain, and the midbrain has 6 top-level subsystems. So The forebrain, incredibly complex, which is basically responsible for cognition and affect, has a very simple highest level, top level interaction configuration. So let me get into now how we think about, how we interpret these subsystems. And for this, we've developed a couple of core hypotheses that are critical for understanding how we're trying to break down this huge amount of data into something that is understandable. And the first hypothesis is that individual clusters, which are distinguished by unique structural connection sets, form stable subsystems with unique functional properties. So a cluster is actually defined by a group of nodes or a group of regions that are more highly connected with each other than with anything else in the network. And so it's really distinguished by a unique set of connections. And the hypothesis is that that set of highly interconnected nodes forms a stable subsystem with a unique functional property or functional properties. So that's a hypothesis. And the other hypothesis is that these structure function subsystems interact within a nested, hierarchically organized global framework. As I just showed you in the previous slide, there's a hierarchical network of these subsystems. So let me give you an example of how we start to think about this. Here's this subsystem 1.1. It's one of the two big subsystems of one of the top level subsystems. And here's what that hierarchy of subsystems actually looks like. So here's the lowest level subsystems down here. And up here is the highest level. Here's subsystem 1.1 up here. You can see this array of lowest level subsystems down here that are arranged in a hierarchy here that ends up at the top. Well, what are in these lower level subsystems here? Here's a great one to start with. And this is a great proof of principle in a way. This subsystem here is the 10 visual cortical areas in the rat. So 10 areas of cortex have been defined actually as visual cortex in the rat. And all 10 of them cluster in a lowest level subsystem here. There aren't any visual cortical areas anywhere else. And interestingly, the part of the thalamus that is associated with this subsystem is the dorsolateral geniculate nucleus. So this makes absolutely perfect sense. And the one right next to it, they're parents of this in this branch over here. This is the auditory perception related cortex. So all 4 of the auditory cortices in rat are clumped together with, interestingly enough, the posterior parietal cortex, temporal association cortex here in the deep layer 6. And over here in thalamus, medial geniculate, suprageniculate, and lateral posterior. So based on a hypothesis here, since we know this is primary auditory cortex, we can hypothesize, sort of as a hypothesis generator, that these 4 other regions over here must be primarily, although not exclusively, auditory in function, because they're in this particular highly interconnected group of regions over here. And you can look at all the other ones, but basically this is the basic idea behind looking at how to actually, if you're interested actually in any one part of the forebrain, let's say, for example, the indusium griseum. Who knows what the indusium griseum does, but it clumps with the dentate gyrus and the medial entorhinal area. This, of course, medial entorhinal to dentate is part of the perforant path in the hippocampus. This would suggest actually that the indusium griseum is probably associated, or maybe an extension of the dentate gyrus. It's a hypothesis, but it is most heavily connected with the medial entorhinal and dentate gyrus. So it's a way of actually maybe getting an idea of what to do experimentally to get at the connections and function of the indusium griseum. And of course, that is here in this higher order branch here with the dorsal hippocampus, okay? And so here we're into a navigation system and so on and so forth. Now, when you follow this up, all the way up, you know, you've got somatic sensory, somatic visceral sensory, somatic motor. You end up actually, there's another extremely interesting area below the ventral, the visual down here. This is the cingulate gyrus over here, basically, with the pre, post, and paracebiculum. This, we're not really sure what the function is, but this correlates to a remarkable extent with the default mode network that people find in human fMRI. So, you know, we almost hypothesize then that this dorsomedial cortical strip over here in this particular cluster here has something to do with cortical executive functions. Here's the input from the anterior thalamus and the mammillary body. So again, this really follows our sort of intuitive pre-knowledge of this part of the brain. But when we start to get into annotating functionally, we can see that it's related to the mammillary bodies and cortical executive function. So when you add all of this up, you end up over here in this S1.1 super second level subsystem over here with the control of voluntary behavior and cognition. Now, when you go down to the other part down here, S1.2, here we get into things like, let's look over here, the accessory olfactory bulb and the parts of the cortex that get inputs from the accessory olfactory bulb and are related really to pheromonal information by and large, including these parts of the amygdala over here. There are a couple of areas in here that we're not quite sure of, but the infralimbic area of medial prefrontal cortex clumps actually in the rat connectionally along with the ventral part of the hippocampus, ventral CA1, with the accessory olfactory bulb, suggesting that actually these parts over here, infralimbic and ventral CA1, have a lot to do with pheromonal sensory motor functions. Down here, just as one other example, here's the AVPV. This is a region that Rich Simmerly, again, studied a lot, and it's part of one of the sexually dimorphic pathways in the rat brain and plays a really critical role in regulating the ovulatory cycle in rodents, at least in rats. And you'll notice it's clumped with Gorski's sexually dimorphic nucleus in the preoptic area, the MPNC here, and with the arcuate nucleus, and with another part of the medial preoptic. So you get into reproductive physiology and behavior. Over here are ingestive behaviors associated with the paraventricular nucleus and so on. Here are suprachiasmatic nucleus, circadian rhythms. And so you end up at the top here with, in this particular one over here, with innate survival behaviors and the integration with physiological responses along with affect. So what about the distribution of these two subsystems, these two top level subsystems? And they could conceivably have a checkerboard distribution in the forebrain, or they actually could form compact, spatially compact arrangements, sort of like continents, let's say. Well, so luckily we have this flat map of the rat central nervous system. Here's the forebrain. And when you plot the blue system, this system, versus the red system, this one, this is what you get. You get an essentially complete spatial segregation of these two subsystems. You can see the blue system has mostly, blue system has mostly dorsal thalamus and most of the cerebral cortex. And the red system is mostly hypothalamus with some of the cerebral nuclei here and a little bit of cerebral cortex, ventral cerebral cortex. And if you plot those on an atlas, you can see the spatial segregation as you go along. Really, here you can see sort of a dorsal lateral blue region and a ventromedial red region. And this boils down actually to an upper level dichotomy, structure-function dichotomy that in a way, in hindsight, actually goes back to the 19th century, and a guy named Edinger, who named the lateral forebrain bundle and the medial forebrain bundle in vertebrate evolution. It's from everything basically from fish to mammals. And what this correlates really with is that the lateral forebrain system, which is the trunk, it is basically the fiber tract running up and down the forebrain, carrying axons up and down, that is actually related to this blue region here. Whereas the medial forebrain bundle is the rostrocaudal fiber tract that is related to the red system here, which is centered in the hypothalamus and ventral parts of the basal ganglion cortex. So if you really take this at a very high level, you've got this lateral forebrain system that is primarily related to the voluntary control of behavior and cognition, and a medial forebrain system that is primarily related to innate survival behaviors and with the integration of physiological responses to these innate survival behaviors and affect associated with these behaviors and physiology. So I don't have time, obviously, to go into all of the evidence that supports this overall theory, but you can read about that in the paper. But this is what this cluster analysis and the hierarchy of subsystems basically leads to, is a high-level look at the overall organization of the forebrain that can then be broken down into finer and finer subsystems. So actually, these subsystems, as you could tell, are interconnected. And I already showed you this interconnections between subsystem 1 and subsystem 2. But of course, then you get into subsystem 1.1 and subsystem 1.2. They're interconnected. And what you can see then, for example, here's the map showing what you can see actually is that the blue and the red, the lateral and medial forebrain subsystems, are interconnected on the same side of the brain, pretty equally on the right and left sides of the brain. And as a matter of fact, this blue cognitive system is connected heavily by commissural connections. Actually, it's not exactly heavy. It's about an 8-to-1 ratio. The internal connections are about 8 times stronger than the corpus callosum connections between the hemispheres. But the hypothalamus part of it, this red part here, very, very weakly interconnected on the right, left sides. And as a matter of fact, there are almost no connections, crossed connections from right to left between the two, between red and pink. So it shows actually quantitatively the interactions between the subsystems. And when you actually stop and think about those hierarchies, there are 166 unique subsystems in this forebrain network, in that hierarchy. If you look, actually there are 166 of these branches in the whole network. So that's really 166 by 166 matrix of subsystem interactions. There are 27,000 possible interactions between these subsystems. But that is opposed to 217,000 possible connections between the nodes themselves. So actually the subsystem level of analysis simplifies thinking about the organization of the forebrain networks a lot. It's still very complicated, but there's a hierarchy in there. And as a matter of fact, you could actually subject this matrix here to cluster analysis if you wanted to, and simplify things down even more. So male versus female, let me just quickly say there are two places in the hierarchy where there are male-female differences. Remember, there are only 4 connections, here and here. But those 4 connections actually have pretty widespread effects on the overall coherence of the forebrain network. And over here in D, what you can see, those 4 connections that are different between males and females, over here, they actually lead to, in D, reordering of the borders of the subsystems over here. So you can see in the female, you have 3 subsystems in this little region right here, whereas in the male, you have 2. And so you have actually the same hubs or the same parts of the brain here, but they're actually connected differently. They're clustered different, which means their parent relationships up in the hierarchy are different. And over in subsystem E over here, actually, you have the same set of subsystems, these 4 subsystems, but they're in a different order. So here's subsystem W, here's subsystem Z across here, and X and Y over here. And so just having these 4 sexually dimorphic connections in this huge network actually leads to significant reordering of the subsystem hierarchy in males and females. And I'll show you just in a second what actual effect that has on the whole network. But before I get there, let me just say that there are some global network features, like what are the hubs and the rich club in the forebrain? And here, it turns out, if you look, there are a small number actually of hubs, of high-level hubs in the forebrain. And look in the cortex over here, first of all, at the hubs. Remember, these are highly connected nodes, the highest, most highly connected hubs in the network. Back here in the temporal lobe, there are 2 of them, the lateral entorhinal area and the perirhinal area. These are in the inferior temporal lobe. And this is actually there's a lot of evidence that this is where Alzheimer's disease, tauopathy, starts in the human brain, in the lateral entorhinal area, perirhinal zone. These are the most highly interconnected parts of the cerebral cortex, actually, the lateral entorhinal and perirhinal area.
Larry, I told you I wouldn't interrupt you, but Dr. Fuster has a question.
Oh, okay. Hi. Oh, I think you're muted.
How about now?
Oh, yeah, I can hear you. Interesting experiments, computational experiments, by doing different matrices. And one would be just what is the difference actually in that huge matrix, the 466 by 466 matrix, between the male and female. Okay, so here we have really just subtracted the male matrix from the female to see what the difference is. And remember, there are only 4 connections that are dimorphic, and yet look at how widespread actually the differences are between the male and female. It's many, many more than 4 connections. In fact, they basically spread out into this whole subsystem here, subsystem 1.1. And, you know, the way to, well, so this, of course, over here is just control. It's one female run versus another, and there are no differences. So this is a way of showing actually how 4 dimorphic connections out of 17,000 can actually have a widespread effect within basically the whole half of the forebrain. But even more interesting, let's computationally almost do something like a complete Alzheimer's lesion. Let's lesion or take out the lateral entorhinal area and perirhinal area from that matrix, take out basically the connections of these 2 regions and see what effect it has on the whole network. So this is the female brain minus the lesion of the lateral entorhinal and perirhinal cortex. It's stunning. It affects virtually every connection in the entire forebrain. The cohesiveness of the entire network is disrupted by the lesion of these 2 areas over here. You know, imagine that you suddenly shut down the intersection between Wilshire and Westwood Boulevard. What effect would that have on traffic in basically the whole UCLA Westwood area? Okay, shutting down one intersection. If you shut it down another huge intersection, say at Santa Monica and Wilshire at the same time, you'd have this disruption of traffic over a really wide area of West LA. And so that's basically what's happening here with these highly, highly connected parts of the brain. And I'm not going to go into the pattern, but you can see basically that there's a very interesting thing here where actually on the contralateral side, there's less coherence in the intact animal and more coherence on the lesion side. And actually the pattern between the male and female is about the same. It's just about the same as it is in the unlesioned animal. Now, if the other thing is, if you lesion, let's say the infralimbic area over here, this is, you know, this is the area in, this actually is the only part of cortex that is both a hub and a rich club over here. This is the part of cortex that Helen Mayberg and her colleagues have been stimulating, deep brain stimulation to relieve the effects of clinical depression. Really has a major, major component of affectivity to it. Well, you can see by lesioning, taking out this one node in the whole network produces these very, very widespread effects throughout the whole forebrain. So it just makes the point that how widespread the effects, the network effects are of influencing one hub in a network. And you know, there's an infinite number of computational experiments you can do to start testing hypotheses and then go in and actually look experimentally at what the results actually are when you do this. We provisionally provided a sign, glutamate or GABA, to every connection in the forebrain. I mean, there are all kinds of caveats to this, but let's just say we did it as a way of starting to think about which are excitatory and which are inhibitory connections in this network. And what you can see here is that in the lateral forebrain system in here, the so-called cognitive system, 16 out of 20 of the subsystems in there exclusively use glutamate as a neurotransmitter. The other 4 use glutamate and GABA. Whereas if you go over the medial forebrain system, essentially all of the subsystems use a combination of glutamate and GABA. So actually this blue cognitive system, the dorsal thalamus cortex system, is almost, the projections between the nodes are almost all excitatory. I mean, it's hardly surprising that you get epilepsy in a system like that if inhibition is interfered with. So our project is about half finished at this point. What does the whole thing looks like? Well, I've shown you the forebrain up here, but you know, in the end, we're trying to get at this entire nervous system with the central nervous system and the forebrain. We think it's going to take 3 or 4 more years to finish that. We call this whole thing here the neurome, as opposed to the connectome, which is the connections of the brain. And, you know, back just for a second to perspective, I found this quote in a British medical journal. It's typical British prose, but it says, "It's not a little remarkable that what has been definitely known regarding the special function of the nervous system has been ascertained within the last 30 years. Well, of course, this was written in 1840, and you know, nothing really has changed in the meantime. But here's what we're faced with right now, and I've shown you this incredibly complex rodent forebrain macro connectome. Okay, it's a 500 by 500 matrix, order of magnitude, with about 250,000 connections. When we get to the rodent macro neurome, to this thing here, that's about 1,000 by 1,000 matrix. Okay, that's 1 million possible connections just in the macro connectome of the rat nervous system. If you go to the rat nervous system at the neuron level, there are about 10 to the 8th neurons in the rat nervous system. So you're looking at a matrix, a 10 to the 8th to 10 to the 8th matrix. It's unimaginable, actually, with 10 to the 16th possible connections between those neurons. And if you're actually thinking about the human brain connectome between neurons, that's in a matrix of 10 to the 11th by 10 to the 11th. That's about a 10 to the 22nd possible connections. What we've got here is a combinatorial explosion, because, you know, the possible number of possible connections is n squared, proportional to n squared, which I've just shown you. But the number of possible combinations of connections, this is what happens during function. When you start to have combinations of functions changing over time, possible combinations of functions are proportional to 2 to the n. Now, you know, it's been estimated that there are only 2 to 266 elementary particles in the entire universe. So when we start talking about the function of the brain at the neuron level and the dynamics changing over time, we're dealing with numbers that are unimaginably big. They're much bigger than the number of elementary particles in the universe. So we absolutely have to come back and get to something strategizing sensibly. And Jeff Lichtman and Josh Sains wrote this wonderful, wonderful article, which I really recommend if you haven't read it, go back. It's called Ohm Sweet Ohm, What Can the Genome Tell Us About the Connectome? They wrote this back in 2008 to counter people's arguments against studying the connectome. The same arguments that were used to counter studying the genome. It was amazing how much resistance there was to studying the genome. And basically the genome strategy was the time-honored strategy for any complex problem. Start from coarse and go to fine. The genome started with a genetic linkage map, then they arranged DNA clones into a physical map, then they divided and sequenced and reassembled the clones, then they filled in the missing details. The same thing is going to happen with the connectome strategy. And the basic idea is start with something that is characteristic of the species at the coarse, gross anatomical level, something that's genetically programmed in the embryo. It's a species level wiring diagram of the brain, and eventually get down to what's specific for individuals at a given time. That's the synaptic organization of the brain. Epigenetic modifications are happening all the time. That's a much more difficult problem. So, 100 years from now, if anybody's still around, they're going to be reading Nature Neuroscience reviews, and they're going to read, it's not surprising that what we know for sure about the amazing functions of the nervous system have been determined in the last 10 years. Nothing changes. But, Cajal, good old Cajal, he said, a great deal remains to be learned about the brain and spinal cord, a task that will take centuries, not years, to complete. And he was certainly true. So here, just in closing, let me acknowledge my two collaborators on this project, Joel Hahn at USC and working with Hong Wei and Olaf Sporns at Indiana University, Mr. Connectome. And finally, here's my favorite picture of Tom Sawyer. Who can forget that raspy, that soft, raspy voice with the soft, great sense of humor at a departmental picnic in Santa Monica at some point with another one of my dear friends, Larry Kruger. So thanks very much for your attention. I'm sorry I went a little bit over, but if we have any time, I'd be glad to answer any questions you might have.